So one of the major discoveries of the 20th century in biology was the discovery of the structure of the DNA molecule, or the elucidation of the structure of the DNA molecule. And that occurred in 1953 when Watson and Crick determined that the DNA had a double helix structure, along the inside of which were these four chemical subunits called bases, or nucleotide bases. And then 4 years later, Crick took that discovery one step further and realized that those chemical subunits on the inside of the molecule were functioning just like alphabetic characters in a written language, or digital characters like the zeros and ones we use in software. In other words, it wasn't the structure of the chemical structure of those subunits, or their molecular weight, or their shape that mattered. What mattered was their arrangement in accord with an independent code that was later discovered called the genetic code. So inside the DNA molecule, what we have is literally information or instructions inscribed digitally or alphabetically, typographically, in a way that provides the information that's necessary to build the important proteins and protein machines that keep all cells alive. So this is a stop-press moment in the history of biology. People have wondered for centuries, why does like beget like? And at least in the case of why we get new proteins that are like the old proteins, the answer is the DNA contains the information for building them, and that's what keeps living cells alive. Well, we're here in Seattle, and we have a lot of great companies here, especially tech companies. And we've got the famous Microsoft company that writes code in the form of software. We also have the Boeing company and other companies that use a technology known as computer-assisted design and manufacture. And that's a technology where information in a digital form is used to direct the construction of mechanical parts or physical systems. So if you're a Boeing engineer, you might sit at a console and write code. That code will go down a wire. It will be translated into another machine code that can be read at a manufacturing arm or center, and then that information will be used, for example, to take rivets and put them on the airplane wing at just the right place. So you have digital information directing the construction of a mechanical system. Something very much like that is going on inside the cell, where the information inside DNA is being used to direct the construction of the proteins and protein machines that are necessary for all cellular life. Natural selection and random mutation don't seem to be a very good explanation for the origin of new genetic information. And the reason for that can be understood by reference to our own experience with digital code, with software. If you're a computer programmer, you know that if you start randomly changing the sections of functional code, you're going to degrade that code long before you ever come up with a new program or operating system. And the same thing applies in the biological case, because the information in DNA is essentially digital or typographic. And because there are far more ways to arrange those A's, C's, G's, and T's along the DNA molecule that will result in gibberish than will result in functional information capable of building a new protein. Inevitably, as random changes accumulate, they're going to find those non-functional combinations. And the process of evolution is going to have, or the process of natural selection is going to have nothing to select. Remember, natural selection can only select for functional advantage. If the code is degraded by random mutations before you ever get to something new and functional, then the evolutionary process is going to terminate. So the natural selection random mutation process is very good for preserving existing function in existing form. It can preserve slight variations on that form, but it can't generate anything fundamentally new. And that's why you have a lot of evolutionary biologists themselves today saying things like natural selection and random mutation can explain the survival, but not the arrival of the fittest. They explain the small scale variations or changes, but they do not explain where major innovation comes from in the history of life. All right, the reason that random changes inevitably degrade the information in a DNA sequence is the same reason that random changes inevitably degrade information in a section of computer code or English text. For every sequence of 12 letters in English language that does convey a meaning, there are 100 trillion other possible ways of arranging those same characters that don't. And the same thing turns out to be true in the DNA protein case. For a modest protein of about 150 amino acids long, for every sequence that produces a functional protein, there are about 10 to the 77th other possibilities that don't. And what that means is then there's a huge search space that has to be searched by the random mutation mechanism to try to find the functional sequences in that vast array of possibilities. And it turns out when you do the math, there isn't enough time, even assuming a 4 billion year history of life on this planet, to have enough replication events, enough copying events to search that space effectively. You're going to end up with a time, even if all of those, every time an organism replicates from the beginning of the first life till now, a new mutation occurred searching for a new protein, you'd only search a tiny fraction of the total number of possibilities. And so it becomes much more likely that such a random search will fail than it is that such a search will succeed. The bottom line is the neo-Darwinian mechanism is just not a plausible mechanism for generating new functional biological information. As it's become clear that the various evolutionary mechanisms do not account for the origin of genetic information, naturally people have wondered, well, what might? And we've proposed an alternative explanation, and that is the idea of intelligent design. And the reason that intelligent design provides a good scientific explanation for the origin of the new information necessary to build new proteins and new forms of animal life is that we know from our experience that information always arises from an intelligent source. Whether we're looking at hieroglyphic inscription or a paragraph in a book or information in a section of software code, or even information embedded in a radio signal. Whenever we see information and we trace it back to its source, it always comes to a mind, not an undirected material process. So the discovery of information at the foundation of life and the discovery that big infusions of information are necessary to explain the origin of new forms of life suggests the activity of a designing intelligence in the history of life. The inference to intelligent design is based on our uniform and repeated experience of cause and effect, which is the basis of all scientific reasoning. We know from experience not only that the neo-Darwinian mechanism and other similar, more recent evolutionary mechanisms that have been proposed have failed to generate the information, the digital and functionally specified information necessary to build new proteins and new forms of animal life. We also know that there is a cause, we know of a cause that does generate functional digital code or digital information, and that cause is intelligence. And we have experience of that in our own realm of software technology or our human experience. In fact, here locally, our hero Bill Gates has said that DNA is like a software program, only much more complicated than any we've ever created. Now, what we know from experience is that it takes a programmer to make a program, to make a computer program. In fact, more generally, we know that whenever we see information, whether it's a software program or a hieroglyphic inscription or a paragraph in a book, whenever we see information and we trace it back to its source, we always come to a mind, not a material process. So the discovery of information at the foundation of life in every living cell of every organism, and the evidence of big infusions of new information coming into the biosphere in the fossil record, both suggest that a designing intelligence has played a role in the construction of the information necessary to life throughout the history of life. And so the inference to intelligent design is not just an argument from ignorance. It's not just we're not just saying that, hey, natural selection and random mutation can't generate new information, or various chemical evolutionary processes have failed to explain the origin of information. We're also saying we know of a cause that does produce information. That cause is mind, and therefore, based on that positive knowledge of cause and effect, we can infer intelligent design as the best explanation, because in fact it's the only known cause of the generation of functional information, especially in a digital or in an alphabetic form. One of the great information scientists of the 20th century said, the creation of new information is habitually associated with conscious activity. That's what we know, and therefore the inference to design is a really strong inference from what we know, both about the facts of biology and about what it takes to generate information. A lot of scientists, because of the influence of materialism, are in a way inured to the reality of their own minds and therefore of minds in general. There's a kind of computer simulation known as a genetic algorithm where evolutionary scientists tried to show how the mutation selection mechanism would work and they do this in a computational environment. They write a little program, they try to get something that simulates mutation and selection to generate a particular sequence of characters, or a phrase from Shakespeare was Richard Dawkins' preferred way of doing it. Methinks it is like a weasel. But in order to get the computer to generate the sequence, Dawkins has to first give the computer the sequence, and then he writes the program, his computer program, in such a way that as different randomized crops of letter strings are generated, the program selects the ones that are closest in function, or that are closest to the future function that he wants. Now, none of the original strings have any linguistic function at all. They don't have any meaning, but the program selects the ones that are closest to the future function that's desired. So he's selecting for proximity to future function, not actual function. Now that's problematic because natural selection selects for functional advantage. It doesn't have foresight to tell you what might eventually result in some future function. So the program that Dawkins generates is not actually analogous to what's going on in biology at all, or how the natural selection mechanism must work. But notice also, the extent to which it does work is entirely a function of his own mind, his own intellect. He gave the program the target sequence. He programmed the computer to select for proximity to future function. So these genetic algorithms invariably end up illustrating the need for mind to generate information. A similar thing happens with what are known as ribozyme engineering experiments. There, chemists attempt to generate RNA molecules that are capable of copying themselves and thus could conceivably get something like natural selection going, because there would be an element of self-replication. The problem is that as the RNA chemists, the ribozyme engineers, have done their work, they've had to sequence the RNA molecules in very specific ways to get any copying capability out of them at all. As it turns out, they've only been able to generate RNA molecules that can copy about 10% of themselves. But even that limited self-replication capability is the product of intelligence, because it was the intelligent agent who essentially, nucleotide base by nucleotide base, determined the informational sequence that the RNA molecule or RNA ribozyme has. So again, what's being simulated? Well, it appears to us that what's being simulated is the need for intelligence to generate information. So far from the idea that intelligent design is the only known cause of information, and that far from the idea that the connection between intelligence and information is only a kind of common sense thing that has no scientific basis, we actually see that the attempt to simulate evolution is providing additional scientific basis for that connection. A lot of people reject the idea of intelligent design because they think that if you invoke the activity of a mind, you're invoking some unintelligible entity of which we have no real knowledge, or an entity, the conscious mind, which is not materialistic. But in fact, I want to argue that we know that we have conscious minds. We know our own consciousness better than we know anything else. In fact, all of our knowledge of the world around us is mediated to us through our senses and comes to us in our minds. So if the mind isn't a real entity, then we don't have any knowledge, let alone scientific knowledge. So mind is presupposed in all scientific inquiry. And getting rid of the mind as an explanatory entity is contrary to everything we actually know. We know from direct introspective activity, better than we know anything else, that we have minds, and we also know from our introspective activity the causal powers that our minds have, what our minds can do. And one of the things we know our minds can do is generate information. I'm doing it right now as we're speaking. So mind is a real entity, it's part of the world, and we've become inured to its reality. but it does really exist, and a good science will open itself up to all that takes place in the world, all the causes that are at work, including mental or conscious causes that are generated by minds.
For the longest time, I believed that Darwinian evolution explains what we saw in biology, not because I saw how it could actually explain it, but because I was told that it did explain it. In schools, I was taught Darwinian biology. And through college and graduate school, I was in an atmosphere which just assumed that Darwinian evolution explained biology. And again, I didn't have any reason to doubt it. It wasn't until about 10 years or more ago that I read a book called Evolution, a Theory in Crisis by a geneticist by the name of Michael Denton, an Australian. And he put forward a lot of scientific arguments against Darwinian theory that I had never heard before. And the arguments seemed pretty convincing. And at that point, I started to get a bit angry because I thought I was being led down the primrose path. Here were a number of very good arguments, and I had gone through a doctoral program in biochemistry, became a faculty member, and I had never even heard of these things. And so from that point on, I became very interested in the question of evolution, and since have decided the Darwinian processes are not the whole explanation for life.
Michael Behe's skepticism derived in large measure from what modern biology has revealed about life's most fundamental unit, the cell.
In the 19th century, when Darwin was alive, scientists thought that the basis of life, the cell, was some simple glob of protoplasm, like a little piece of jello or something that was not hard to explain at all.
This perception didn't really change too much until the early 1950s. But in the last half century, our knowledge of the cell has just exploded.
Today, powerful technologies reveal elaborate microscopic worlds. Worlds so small that a thimbleful of cultured liquid can contain more than 4 billion single-cell bacteria, each packed with circuits, assembly instructions, and miniature machines, the complexity of which Charles Darwin could never have imagined.
At the very basis of life, where molecules and cells run the show, we've discovered machines, literally molecular machines. There are little molecular trucks that carry supplies from one end of the cell to the other. There are machines which capture the energy from sunlight and turn it into usable energy. When we look at these machines, we ask ourselves, where do they come from? And the standard answer, Darwinian evolution, is very inadequate in my view.
One machine particularly attracted his attention.
I remember the first time I looked in a biochemistry textbook and I saw a drawing of something called a bacterial flagellum with all of its parts in all of its glory. It had a propeller and the hook region and the drive shaft and the motor and so on. I looked at that and I said, that's an outboard motor. That's designed, that's no chance assemblage of parts.
Behe's reaction was not surprising, for the molecular motors that drive bacteria through liquid each depend upon a system of intricately arranged mechanical parts. These parts come into focus when portions of a cell are magnified 50,000 times. Biochemists have used electron micrographs like this one to identify the parts and three-dimensional structure of the flagellar motor. In the process, they have revealed a marvel of engineering on a miniaturized scale.
Howard Berg at Harvard has labeled it the most efficient machine in the universe. These machines, some of them are running at 100,000 RPMs and are hardwired into a signal transduction or sensory mechanism so that it's getting feedback from the environment.
And even though they're spinning that fast, they can stop on a dime. It only takes a quarter turn for them to stop and shift directions and start spinning 100,000 RPM in the other direction.
And just like outboard motors on motorboats, it has a large number of parts which are necessary for the motor to work.
The bacterial flagellum, 2 gears, forward and reverse, water-cooled, proton motive force. It has a stator, it has a rotor, it has a U-joint, it has a drive shaft, it has a propeller, and they function as these parts of machines. It's not convenient that we give them these names. That's truly their function.
Since its discovery, scientists have tried to understand how a rotary motor could have arisen through natural selection. As yet, they have failed to offer any detailed Darwinian explanation. To see why, we must understand a feature of molecular machines known as irreducible complexity.
Irreducible complexity was coined by Mike Behe in describing these molecular machines. Basically what it says is that you have multi-component parts to any given organelle or system in a cell, all of which are necessary for function. That is, if you remove one part, you lose function of that system.
The idea of irreducible complexity can be illustrated by a familiar non-biological machine, a mousetrap. The trap is composed of 5 basic pieces: a catch to hold the bait, a strong spring, a thin bent rod called the hammer, a holding bar to secure the hammer in place, and a platform upon which the entire system is mounted. If any one of these parts is missing or defective, the mechanism will not work. All components of this irreducibly complex system must be present simultaneously for the machine to perform its function, catching mice. Irreducible complexity also applies to biological machines, including the bacterial flagellar motor.
All told, there are about 40 different protein parts which are necessary for this machine to work. And if any of those parts are missing, then either you get a flagellum that doesn't work because it's missing the hook or it's missing the drive shaft or whatever, or it doesn't even get built within the cell.
In evolutionary terms, you have to be able to explain how you can build this system gradually when there's no function until you have all those parts in place.
But could Darwin's small, favorable variations have produced a bacterial flagellum? Some scientists doubt the possibility.
How could something new, like a bacterioflagellar motor and all of the components that go with it, how could it develop out of a population of bacteria that don't have that system? When each change, according to Darwin's theory, has to provide some kind of advantage.
Imagine such a scenario early in the Earth's history. An evolving bacterium somehow develops a tail and perhaps even the pieces necessary to attach it to the cell wall. Yet without a complete motor assembly, this innovation would provide no advantage to the cell. Instead, the tail would lie immobile and useless, invisible to natural selection, which by definition can only favor changes that aid survival.
The logic of natural selection is very demanding. Unless the flagellum mechanism is completely assembled and actually works, natural selection simply cannot preserve it. It cannot be passed on to the next generation.
The important thing to realize about natural selection is it selects only for a functional advantage. In most cases, natural selection actually eliminates things that have no function or that have a function that harms the organism. So if you had a bacterium with a tail that didn't function as a flagellum, chances are natural selection would eliminate it. The only way you can select for a flagellum is if you have a flagellum that works, and that means you have to have all the pieces of the motor in place to begin with. So natural selection can't get you the bacterial flagellum. It can only work after the flagellum is there and operating.
In 1996, Michael Behe published a book titled Darwin's Black Box. In it, he argued that natural selection, Darwin's designer substitute, could not explain the origin of the bacterial flagellum or any other irreducibly complex biological system. Instead, Behe concluded that the integrated complexity of these systems pointed to intelligent design. Darwin's black box created immediate controversy. Over 75 publications, including some of the world's leading newspapers and scientific journals, reviewed the book. Some scientists praised Behe's work, while others dismissed it as unscientific and religiously motivated. Behe's critics also insisted that he had underestimated the power of natural selection. They argued that the flagellar motor could have been constructed from parts used to build simpler molecular machines, like this needle-nose cellular pump. If the components of the pump already existed, they could have been preserved by natural selection even before the bacterial motor arose. This theory is called co-option.
It's essentially saying that evolution or natural selection at some point was able to borrow components of one molecular machine and build a new machine with some of these components.
Scott Minnick has studied the flagellar motor for nearly 20 years. His research has led him to challenge the co-option argument.
With a bacterial flagellum, you're talking about a machine that's got 40 structural parts. Yes, we find 10 of them are involved in another molecular machine, but the other 30 are unique. So where are you going to borrow them from? Eventually, you're going to have to account for the function of every single part as originally having some other purpose. So you can only follow that argument so far until you run into the problem of you're borrowing parts from nothing. But even if you concede that you have all the parts necessary to build one of these machines, that's only part of the problem. Maybe even more complex, I think more complex, is the assembly instructions. That is never addressed by opponents of the irreducible complexity argument.
Studies of the bacterial motor have indeed revealed an even deeper level of complexity. For its construction not only requires specific parts, but also a precise sequence of assembly.
You've got to make things at the right time. You've got to make the right number of components. You've got to assemble them in a sequential manner. You've got to be able to tell if you've assembled it properly so that you don't waste energy building a structure that's not going to be functional.
Building a molecular machine has been compared to the construction. So it is with the construction of a flagellar motor.
You build this structure from the inside out. You are counting the number of components in a ring structure or the stator, and once that's assembled, there's feedback that says, okay, no more of that component now. A rod is added, a ring is added, another rod is added, U-joints added. Once the U-joints at a certain size and a certain degree of bend, about a quarter turn, that's shut off, and then you start adding components for the propeller. These are all made in a precise sequence, just like you would build a building.
To build a motor correctly requires a complex system of machines that coordinate the timing of the assembly instructions. But how could natural selection construct such a system?
The co-option argument doesn't explain this. You see, in order to construct that flagellar mechanism, or tens of thousands of other such mechanisms in the cell, you require other machines to regulate the assembly of these structures. And those machines themselves require machines for their assembly.
If even one of these pieces is missing or put in the wrong place, your motor isn't going to work. So this apparatus to assemble the flagellar motor is itself irreducibly complex. In fact, what we have here is irreducible complexity all the way down.
We know a lot about the bacterial flagellum. We still have a lot to learn, but we know a lot about it. And there's no explanation for how this complex molecular machine was ever produced by a Darwinian mechanism.
150 years ago, scientists did not know about irreducibly complex molecular machines. Yet Charles Darwin anticipated the difficulty that systems such as these could pose to his theory.
If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down.
These are tiny molecular machines, and they are doing this inside your body right now. To understand why, we have to zoom out. Every day in an adult human body, 50 to 70 billion of your cells die. Either they're stressed or damaged or just old. But this is normal. In fact, it's called programmed cell death. But to make up for all these lost cells, right now, billions of your cells are dividing, essentially creating new cells. And that process of cell division, also called mitosis, well, it requires an army of tiny molecular machines. So let's take a closer look. DNA is a good place to start, the double helix molecule we always talk about. This is a scientifically accurate depiction of DNA created by Drew Barry at the Walter and Eliza Hall Institute of Medical Research. If you unwind the two strands, you can see that each has a sugar phosphate backbone connected to the sequence of nucleic acid base pairs, known by the letters A, T, G, and C. Now the strands run in opposite directions, which is important when you go to copy DNA. Copying DNA is one of the first steps in cell division. Here, the two strands of DNA are being unwound and separated by the tiny blue molecular machine called helicase. Helicase literally spins as fast as a jet engine. The strand of DNA on the right has its complementary strand assembled continuously, but the other strand is more complicated because it runs in the opposite direction. So it must be looped out with its complementary strand assembled in reverse, section by section. At the end of this process, you have two identical DNA molecules, each one a few centimeters long, but just a couple nanometers wide. So to prevent the DNA from becoming a tangled mess, it is wrapped around proteins called histones, forming a nucleosome. These nucleosomes are bundled together into a fiber known as chromatin, which is further looped and coiled to form a chromosome, one of the largest molecular structures in your body. You can actually see chromosomes under a microscope in dividing cells. Only then do they take on their characteristic shape. Otherwise, the DNA is more strewn inside the nucleus. The process of dividing a cell takes around an hour in mammals, so this footage is from a time-lapse. You can see how the chromosomes line up on the equator of the cell. Now when everything is right, they are pulled apart into the two new daughter cells, each one containing an identical copy of DNA. Now, as simple as this looks, the process is incredibly complicated and requires even more fascinating molecular machines to accomplish it. So let's look at a single chromosome. One chromosome consists of two sausage-shaped chromatids, containing the identical copies of DNA made earlier. Each chromatid is attached to microtubule fibers, which guide and help align them in the correct position. The microtubules are connected to the chromatid at the kinetochore, here colored red. The kinetochore consists of hundreds of different proteins working together to achieve multiple objectives. In fact, it's one of the most sophisticated molecular mechanisms inside your body. The kinetochore is central to the successful separation of the chromatids. It creates a dynamic connection between the chromosome and the microtubules. For a reason no one's yet been able to figure out, the microtubules are constantly being built at one end and deconstructed at the other. While the chromosome is still getting ready, the kinetochore sends out a chemical stop signal to the rest of the cell, shown here by the red molecules, basically saying this chromosome is not yet ready to divide. The kinetochore also mechanically senses tension. When the tension is just right and the position and attachment are correct, all the proteins get ready, shown here by turning green. At this point, the stop signal broadcasting system is not switched off. Instead, it is literally carried away from the kinetochore down the microtubules by a dynein motor, that's the walking guy. This is really what it looks like. It has long legs so it can avoid obstacles and step over the kinesins, molecular motors that walk in the opposite direction.
Personally, I'm astounded by these tiny molecular machines, how they're able to routinely and faithfully execute their functions billions of times over inside your body at this exact instant. I'm also amazed by the scientists who were able to work out how this happens in such detail that we could create realistic depictions of them like you saw in the animations in this video. But perhaps the most amazing thing is just how much is left to be discovered, like figuring out how exactly the chromatids are pulled to opposite ends of the cell. There is still so much that we don't quite know. You know, what I find exciting is that in science fiction, for decades, we've been writing about tiny nanobots that will be injected into our bloodstreams that can heal us. But what this suggests, the existence of these tiny molecular machines inside us, it suggests that there isn't a physical limit that would prevent that. And so I think it's pretty likely that in future, we will be able to develop our own tiny molecular machines that will be able to repair our bodies better than they can repair themselves.
When applied to the origin of life and the random formation of large biomolecules, probability theory clarifies the limitations of chance as a creative agent on the primordial Earth. For example, what are the odds a single protein could form exclusively through the blind interactions of chemistry? Our target is one smaller than average molecule made from 150 amino acids, each aligned to ensure a folded chain. Researchers have calculated that on the ancient Earth, the probability of success was 1 chance in 10 to the 164th power. That's one correctly sequenced protein chain for every 100 million trillion 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 failed attempts. But despite these enormous odds, some theorists argue that given enough time, anything is possible. Okay, let's test the validity of this opinion. We'll begin by establishing an ideal environment for chemical evolution, an imaginary world that will provide chance with every opportunity to succeed. First, we stock the oceans to capacity with amino acids. That means all the atoms on Earth, including its entire supply of carbon, nitrogen, oxygen, hydrogen, and sulfur, are available to form 10 to the 41st complete sets of the 20 types of amino acids used to build proteins. Then we'll alter the laws of nature to protect these building blocks from the destructive rays of ultraviolet light and chemical contamination in the primordial soup. Now let's turn the chemistry loose and see what happens. The amino acids start bonding furiously. In our experiment, an entire chain of 150 units self-assembles in only 1 second. Since all 20 types of amino acids are available, at the majority of sites, there is a 5% or 1 in 20 chance the correct molecule will align in the chain. If the sequencing is incorrect, the chain is immediately destroyed and a new assembly begins. Throughout the planet, 6,000 million billion trillion trillion attempts will take place every minute. That means in 4.6 billion years, the oldest estimated age of the Earth, the number of chains that don't fold will exceed 10 to the 58th power. It's a staggering total, but nowhere near 10 to the 164th. The trials necessary, on average, to build a protein of 150 amino acids by chance. So if undirected chemistry can't produce our coveted molecule during the entire history of the Earth, then how much time would have been needed? To find out, let's take a road trip. We'll start by erecting a bridge that spans the diameter of the observable universe, a distance of more than 90 billion light years. Then we'll place an amoeba on one end of the bridge. This single-celled organism will travel at the breakneck speed of 1 foot per year. It's off. While we wait for one protein to form by chance, the amoeba slides along for more than 5 billion years to cross the entire universe and then return. But this race is just getting started. The amoeba takes off again, successfully reaches the far side of the cosmos, then heads back home. Yet not even one functional protein is anywhere in sight. For the next trip, we'll add a payload, a single atom. After inching its way another 500 billion trillion miles, the amoeba drops off its cargo and returns for more. Will it get back before our lucky protein forms? Absolutely. In fact, it will complete another 10 round trips, then 20, 100, 1,000. And there's still no sign of a usable molecule. The amoeba continues making round trips until it has hauled off every atom on Earth. Then, all of the atoms in our solar system. Then, every planet and star in the Milky Way galaxy, one atom at a time. In fact, as we wait for one protein to self-assemble, the amoeba has so much time that moving at just 1 foot a year and carrying 1 atom per round trip, it will transport the entire universe more than 56 million times. That's how long it would take for chance to build one functional protein.
Now, suppose against all odds, chemical evolution produced our single functional protein. Would we have life? No. We'd have one protein, just a lifeless arrangement of amino acids. The simplest living cell we know has more than 300 different proteins. But proteins are only part of the story when you consider any actual cell. Remember, you're going to have carbohydrates, complex sugars, nucleic acids, DNA and RNA, lipids, a whole variety of different chemicals which jointly constitute the living state. Those bits and pieces all have to be brought into the same microenvironment at the same moment in time.
Each chemical building block must then be assembled and organized into the network of molecular machines that will control every facet of life.
If we can appreciate exactly how hard it is to produce one molecular machine using nothing except atoms and energy, we can see that there's a profound problem, because once you have one molecular machine, you don't have a living thing. These molecular machines need other molecular machines. And even if nature was capable of producing all the molecular machines necessary, that still wouldn't be enough. They have to all be together, all in this tiny little membrane-bound space that we call a cell.
From my understanding of what it takes to make a cell, it has to happen all at once. You can't do it one bit at a time, because everything works together in a causal loop. The higher level of organization transcends the pieces. The spatial organization in the cell requires that molecules end up in the right place at the right time. The DNA is copied into RNA. The polymerase that does the copying has to find the right spot in the DNA to start copying. The RNA has to somehow hook up with ribosomes, which have to be in a particular place. And the proteins then that are made have to be going to a particular place. That's an awful lot to account for by random chance.
The probability that you would get them in the same space at the same time becomes beyond unimaginable. And the probability that you would get them within a membrane enclosure, like a cell, is the next best thing to impossible.